At this point, you've most likely watched X-Men or something in the Marvel universe with Avengers, and this focuses in on mutations. So all of those superpowers are mutations, and that whole society is very anti-mutant, whereas in reality, we are all mutants in some way. We all have genetic mutations, and they're not necessarily a bad thing. So in this lesson, we're going to learn about what mutations are, what the specific types are, and how they can affect us. So we're on page 81 in our handbook, and you've got that crazy sentence that says, what happens when protein synthesis goes wrong? That sentence itself is a mutation. And you'll learn later that this is a frame shift mutation, which we'll talk about in a little bit. So let's start with first what a mutation is. A mutation is a change in the original DNA sequence, and that can lead to a change in the amino acid sequence. So when we talk about a change in the original DNA sequence, we're talking about the order of the bases, the A's, T's, C's, and G's. If you change that order, you can change your RNA, which can change the amino acid order. And a mutation can happen when that original DNA is not copied right during replication, which remember happens in the S phase of interphase, or it can happen during protein synthesis. These mutations can be spontaneous, meaning they happen randomly, or they can be caused by things such as radiation, like sunlight, or exposure to certain chemicals. That's why some chemicals are called radioactive. We call these things mutagens because they can cause mutations. And that result of a mutation is normally a change in your amino acid sequence, the order of the amino acids that build a protein. Then that protein that's made either might not be made or it might be defective, meaning it doesn't work right, because when you change that order, you mess up the shape of a protein. And remember, proteins are shape specific. And it's important to remember, mutations that are in your sex cells, that egg and sperm, those are the ones that you can inherit. So if you get skin cancer, that's your skin cells mutating. Your baby is not going to have skin cancer because your skin cells are somatic. However, if your egg or sperm is deformed and has a mutation, then you're going to pass that mutation on to your baby. So only gametes can result in changes in the baby. There are two types of mutations, and take a moment to fill in why does a change in DNA mean a change in the protein. And remember, your main idea here is when you change DNA, the mRNA changes, meaning amino acids change. So protein could not work right or not work at all. Okay, so let's get into those types of mutations. We have one kind is called a point mutation, meaning it happens only at one point, one base. They're also known as a substitution because one letter is substituted with another. Remember, the letters represent those bases. So for example, if you have an A, it's replaced with a C. So if you see this example here, if I start with Gattaca, the original sequence is Gattaca. If I look here, this says Gagtica. This is my mutation. This G substituted where the T was. So that is a point mutation because it only happened at one location. So let's get into an example of what could happen here. So a point mutation, if it happens on a gene, can result in the change of just one amino acid in a protein. Because remember, we read 3 letters at a time. And the big disease that happens with this is sickle cell anemia, which is caused by a point mutation that causes your red blood cells to change shape. Let's look at an example here of a point mutation. So first thing is we have our original DNA, which is TAC, GGG, ATC. First, you would find your mRNA. Remember, going from DNA to RNA is transcribing. This happens in the nucleus. So that T would become an A, That A would become a U, then G, C, 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 U, A, G. Not Y, A, G, U, A, G. I then use the amino acid chart to figure out what my amino acids would be here. So my first codon, my first three letters are A, U, G. So my first base is A, my second is U. So that means I'm narrowing down to this box. And then I'm looking for G, my third base. So I go over, and that means my amino acid here is MET, M-E-T. Remember, that's the start codon. So MET. My next codon is C, C, C. So again, first base is C, second is C. So I'm now in this box over here. Everything in this box is pro, but if I look third base G, it would line up with pro. So my next one is pro. That line there represents my peptide bond between those two amino acids. My last is UAG. So if I look, here's U, A. If I go down G, that's my stop codon. So my protein here is just made of the start, pro, and stop. So that's what my normal should be. Now, you have a mutated mRNA, or mutated DNA. So my mutation here is a point mutation because here, there's the letter A instead of the letter G over here. So there was one point where it was different. So now I transcribe my mRNA, AUG, G, C, C, U, and UAG. So my first amino acid is still going to be MET because it's AUG. So I still have my start protein, my start codon. But my next one is CCU. If I go over here, let's choose a different color. We've got C, C, so I'm in this box, U. CCU also codes for pro. And then UAG is still stop. So in this case, my mutation isn't a bad thing because it still codes for the same exact protein. Sometimes, however, your mutation will code for a different protein, and then that's where you've got issues, such as sickle cell anemia. So in sickle cell anemia, if you take a look, this is your original DNA sequence, and normally if you have a normal code for hemoglobin, you've got the amino acid glutamic acid being coded, and you have these normal donutty red blood cells. However, if you have sickle cell, instead of this T here, you have an A, you have a point mutation, and that causes valine to be made. This one little change in the amino acid misshapes your red blood cells. Remember, your blood cells carry oxygen. That means they can't carry oxygen right. Oxygen doesn't get to your cells. You can't do respiration very well. Bad stuff. Because remember, respiration is how you make ATP. If you're not making any ATP, you've got no energy, you're going to be tired all the time. All in all, just a really bad thing based on one little point mutation, one change in your DNA base sequence. The second type of mutation is called a frame shift mutation. Called because it literally shifts your reading frame. Remember, you read 3 at a time with your codons. This is when you insert, add a letter, or delete, get rid of a letter, and it messes up your entire reading frame. So all of your amino acids end up changing. These tend to be the most damaging because you're completely changing what the protein is, so it just wouldn't work correctly. So again, you're either adding a base or getting rid of a base. So this example here, We're going to go through an example and then I'll talk about the effects of one. So if our original DNA is GAT-TAC-ATT, that would make my original mRNA CUA, AUG, UAA. Remember, that was transcription I just did happening in the nucleus. Now I'm going to translate that amino acid. This is happening on the ribosome. So C is my first base, U, A is LEU. That bond, remember, is my peptide bond. Then I have AUG, which is MET. Then I have UAA, which is step. Now, I have a mutation. So over here, you see where this TAC is? I have TAA, then C. So an A was inserted as my mutation. So now, if I look at my mRNA, I have transcribing it CUA, AUU, GUA, and then this random A floating around that obviously isn't going to code for anything because it's not a codon, it's not 3. So now my amino acids, if I look up CUA, there's CUA, that's still LEU. Got my peptide bond. Now I have AUU, AUU. That means ILA is my second amino acid instead of MET. Now I have AUU was ILE. Now I have GUA. GUA is val. So instead of LU-MET stop, I've got L-ILA-val. This is a completely different amino acid sequence. This protein is not going to work at all. Whatever that trait is isn't going to happen. It could be a disease. So the effect of a frame shift will depend on the location of where you added the base or deleted it. If this mutation happens earlier, like at this G at the beginning, more amino acids will be changed and the more harmful the mutation will be. If it's towards the end, it might just change the shape a little bit, but it won't be as big of a deal. The big disease to know about this is Huntington's disease. Huntington's disease is when your nervous system starts functioning poorly, and this is due to the insertion, the adding of several bases that changes the reading frame. So again, these are typically really bad, not good. All right, so now we're on page 82. Take a couple of minutes to check yourself and get a stamp when you are done.